Welcome to BIOS Frontier Science. Today, we're
Yep.
incredibly excited to welcome Jean Yeo, Professor of Cellular and Molecular Medicine at the University of California, San Diego. To help host this episode, I'm joined by my colleague, Drew Yasher. To kick things off, Jean, can you share a brief personal intro with us?
Sure. Thank you for the opportunity to tell you about myself and my work. So I was originally born in Singapore. I moved to the US to pursue my undergrad education at the University of Illinois Urbana-Champaign in chemical engineering and economics. Then I returned to Singapore and served in the military. So I was a naval officer for a year plus and did all the whole basic boot camp before that. And after the military, I decided to apply to grad school and was fortunate to be able to go to Boston, where I completed my degree, my Ph.D. in computational neuroscience, with under the mentorship of Chris Birch, Tommy Poggio, and had a chance to work a lot with Phil Sharp there. And right after I defended, like that same afternoon, I said, well, I'm going to go learn more neuroscience, but focus on developing new models, cell culture models for diseases, and decided to pursue a fellowship at the Salk Institute with Rusty Gage. And San Diego is a hard place to leave. So after being at the Salk for a couple of years, I decided to start my own lab across the street at UC San Diego, where I've been ever since, and have been having a great time, mentoring students and postdocs and building up a research program and understanding the basic mechanisms of RNA processing and trying to use what we learn to develop, paradigms for therapeutics or developing new tools that we hope the community can utilize.
Oh, and thank you for that background. Before we dive too deeply into your current work, your lab, and everything you've built, we'd love to maybe take a step back and also ask what brought you to bio and really healthcare research more broadly?
Right. So I was, in undergrad, as I mentioned, I was chemical engineering, undergrad, and really thinking about tools and methods development. I wasn't really thinking a lot about biology, but I had the opportunity to intern at a early stage company then, Affymetrix. And Affymetrix was one of the pioneers in developing these gene chip arrays where you can measure thousands of genes, or the expression of thousands of genes, right, in parallel. And I was really there to think about the manufacturing of the chips, right, being a chemical engineer. But then I realized that the power of the platform was in really understanding biology at a very different scale, right? Prior to that, folks would do gene expression measurements of one or a dozen genes at the same time, but really not at this different scale. And so I got really, and how do you start developing a systems perspective, or what is now, I guess, called systems biology, but then a systems perspective of how genes are controlled and how cells behave. And so this sort of that fermented in my head for many years, even when I was back home in Singapore in the military. And at the end of that, I said, well, I'm very interested in understanding human biology. I'm super interested in understanding gene expression, but I wanted to apply and develop computational methods to this new area, right? And at the time, the human genome, the draft genome, was about to be published, and suddenly I felt, at least for me, I felt like the life sciences field started moving towards a digital age, right, where data was coming fast and furious at a whole different magnitude than it had before. And so I decided to hone my expertise in high throughput at that point, data analysis, right, and developing a computational perspective in in trying to understand biology and gene expression. So that got me interested in basically molecular biology, computational biology, evolution, and then moving ahead, I sort of we need to put our findings in the context of cell biology, organismal biology, and then slowly moving towards more thinking about diseases. Can we understand development? And before I knew it, I'm a full-blown sort of biologist now, right? And so it's been an interesting journey of I think accidents, right, that have brought me to this area. Yeah.
And I think a lot of the best careers are developed, not always, but a lot of the best careers are developed in sort of a winding path. And I say this as someone who was originally a chemical engineer turned bioengineer.
There you
go. Might also just be personal resonance. But, and you've started to touch on this, but your lab certainly focuses on a variety of subjects at the intersection of these disciplines that include a hybrid. You're talking about data and computation. And so they include a hybrid of computational and experimental RNA biology, neurobiology, all driving towards things, not only developing that mechanistic understanding, but also RNA therapeutics, as you pointed to earlier. So before diving deeper into specific projects, one question we always like to ask our guests, throughout your career, have you had sort of a North Star, a common thread, if you will, that ties your work together, especially given all these exciting areas you're pursuing?
Yeah, I think I'm by nature a very curious person, and got very interested in trying to understand how do you sort of transfer information from one system to another system, right? And one would argue one of the most interesting forms of information transfer or it's coming from the genome, right? You have these 3 billion bases, and that somehow that information is being transferred at some point all the way to proteins and functional non-coding RNAs, and then they form complexes and so on. So this degree of complexity is fascinating to me, right? And I've been stayed very curious about how this happens, right? And decided to pick a slice, because it's hard to study everything under the sun in that area. But my slice is trying to understand how this information encoded in the genome become, how is it read and interpreted, right? When RNAs to generate RNA, and then how does RNA get processed, right, and recognized by signals in the cell or proteins in the cell, and then modulated into different versions of that same information, and how the information is transferred on. So sort of information movement and transfer has always been very interesting to me.
And interesting to the rest of us, which is why we're so excited to have you joining today. And with that, to dive a little bit deeper, I'll pass it over to Drew to discuss a little bit more about RNA binding proteins, our first topic. Drew?
Yeah, thanks so much, Chris. And thank you, Gene, again for joining us. We're so excited to dive into our first topic here. And as Gene, as you mentioned, your lab's major focus lies in the understanding how gene expression is controlled at the RNA level to maintain proper functioning of cells during development of aging. We really can't wait to dive deeper. To really just start here, to provide a bit of context for our audience, can you share more about the history of the RNA expression field?
Sure. Again, we I guess we make our discoveries and stand on the shoulders of giants. And so I was, again, very grateful to be able to do my doctoral work with Chris Birch and Tommy Poggio. And so Tommy comes from a machine learning, statistical learning theory sort of field, and Chris came from a sort of computational biology and RNA field. And through them, I also worked very closely with Phil Sharp. And Phil Sharp obviously was awarded the Nobel for discovering RNA splicing, the concept of split genes, right? And so for me, I mean, of course, the RNA field began, way before that, right? You had the discovery that mRNA, the triplet code of mRNA, is critical for generating what we call proteins, right? And so that was a Sidney Brenner's discovery. among many others. And so it's, obviously it's a field that's been around, but when I started in grad school, I was very interested in again how information at the sequence level in transcribed RNA are recognized and processed. And at that point in time, one of the most interesting questions was how do splice site signals, right, get recognized, and then introns are then removed, right? So you have exons that are then placed together to generate a messenger RNA, except that there are many different versions, choices that can be made. And so you can make different forms, isoforms of mRNAs. And so this process of splicing and alternative splicing was very fascinating to me, right? And at that point, I think there were, the genome was just out, right? You have folks aligning express sequence tags, right, to the genome to try to identify where the splice sites were. And in Chris's lab, I spent quite a fair bit of time trying to understand, what is a strong splice site, what is a weak splice site. I built algorithms in the area, and very happy that some of my algorithms are still being used to date. And from that understanding, we were trying to derive rules for how different sequence motifs within pre-messenger RNA is then recognized to then choose this exon on the other exon to generate different isoforms. And so that was sort of, my introduction to the RNA field through one of the lenses, right? And so it was RNA splicing, but it gave me a really a strong foundation in thinking about signals within RNA sequence, and then ultimately also structure that could be read or recognized, right, by proteins, right? And so as we moved along, or as I moved along my career, I started thinking more about, what are the proteins that then bind these sequence motifs, and then, of course, when I started my fellow position at the Salk, there were maybe a dozen or so labs in the world that studied a dozen or so RNA binding proteins, and many of them are started to have very interesting functions, right, in neurodevelopment and in cancer. And I remember when I transitioned to UCSD and joined the Department of Cellular and Molecular Medicine, one of the questions my chair asked me was, which RNA binding proteins are you going to focus on? At that point, there were maybe a couple 100, 300, 500 RNA binding proteins. They were annotated, and a dozen or so RBPs that were being studied at great detail. I actually said I would like to study all of them, right, and discover new ones and identify new functions for many of these new ones, and potentially develop therapeutics to some of these RNA-related disorders, by understanding RNA binding protein biology. And we've been able to make strides in towards this direction over the last decade now. So to date, right, you have, 2,500 to maybe 4,000 to 5,000 proteins and that are predicted to be RNA binding proteins. We have been able to generate and characterize antibodies for more than 1,000 human RNA binding proteins. In my lab, we've been able to map the binding sites for over 200, now at this point probably 300 RNA binding proteins, and understand their patterns of interaction and molecular mechanisms of action actually on their target genes across a couple of different cell lines. And we've been able to try to use them now as engineering tools, not just understanding their basic normal functions, but can you develop tools from them? And in different diseases where they are misregulated, we are trying to correct the diseases, right? Think about ways to dig deeper into the mechanisms of how dysregulation of RBPs causes neurodegeneration or autism. So it's been a fun journey from a very basic perspective still, and trying to help the community in thinking about translating some of this basic understanding to other areas. Yeah.
think it's a fantastic introduction, Gene. And just to start here and build off this, your lab is really focused on how RNA binding proteins, RBPs, influence RNA processing as regulators and their vital potential as targets in therapeutics. As you've described, with the increasing number of proteins that are now classified as RBPs, and starting the variation in diseases that they seem to cause, fundamental questions about how RBPs function, which RNA targets they interact with, and how they regulate gene expression still remain really unsolved. I'd love to just kind of take a step back before we jump into your own work personally, and just ask directly, what are RBPs and why are they so significant?
RBP is a good working definition, right? Because it's it states RNA binding, right? So basically, if a protein can bind RNA, it's an RNA binding protein, right? And so that's a big sort of catchphrase for many regulators of RNA processing, right? Which includes splicing factors, RBPs that bind to pre-mRNA to modulate RNA splicing or modulate, intron retention, for example. And that also includes RNA binding proteins that modulate RNA stability. Often they bind the 3' untranslated regions of genes to change the localization of the RNA or recruit factors that lead to decay of the RNA or degradation. This also includes RNA binding proteins that are involved in translation initiation and elongation, and the recognition of stalled ribosomes on RNAs. RBPs also include the ribosome actually as complex RNA protein machinery, as well as the spliceosome, another complex RNA protein machinery. And I think the way I think about RNA binding proteins are that many of them are likely to be functional in some context, in some cell type, and still it remains to be seen, what they all do, right? So we know that RBPs can recognize specific sequences, but they're typically very degenerate. So they may involve other proteins that bind them to help specify the interaction with RNA. We also know that RBPs can recognize structure in RNA and that they can be recruited, as part of the transcriptional machinery onto RNA, right? And so our working definition is a broad one, or whatever proteins that can bind RNA. And what we should really be moving towards is trying to understand which ones are functional and what are the functional RNA targets in specific contexts, right? And so that really requires accelerating, the pace of RBP RNA biology or RNA biology as a whole by describing, developing technologies that are robust and scale, and ideally at some point, miniaturized and automated, and so we can, at the level, at single cell level resolution, at subcell level resolution, to then really change the pace really of how we can understand this RBP RNA biology. And the reason why this is really important is because mutations in the binding sites of the structure, RNA structural elements that are recognized by RBPs, that can lead to RBPs not being able to bind or are trapped in these RNAs, right? These sequences, and that leads to a variety of different human diseases. And so, for example, you have myotonic dystrophy that is due to expanded RNA repeats that sequester and trap RNA binding proteins away from their cognate roles as RNA splicing factors. You have defects in or missing components of splicing the spliceosome, for example, that may lead to motor neuron degeneration. You have diseases like Fragile X tremor-associated syndrome, which may be also due to an RNA toxicity. Huntington's disease may also be viewed as a disease where you have an RNA component and a protein component. And of course, many forms of cancer, you have dysregulation of the RNA binding protein level, right? and these RNA binding proteins may act as safeguard mechanisms to enable the tumor cells to initiate, to migrate. And so trying to understand this interaction between RBPs and the RNA targets in the correct context matters quite a bit in first of all understanding that context and the biology, right? The phenomenon, be it neurodifferentiation or degeneration or tumor cell proliferation or migration, right? At the same time, understanding the context and the mapping between RBP interactions allows us to build new tools as well, right? Because then you can take pieces of RNA binding proteins, can you then develop new ways of recognizing RNAs they don't normally recognize, right? Or new ways to probe the life cycle of RNA to understand how they're impactful in different diseases.
That's fantastic. Your lab in particular has pioneered a number of techniques to increase our understanding of RBPs and their function. One quick thing to dive into this as well, just as an overview of the ecosystem overall. Could you tell us more about the underlying technologies enabling the development and assessment of these proteins today?
Right. So we've been very focused in trying to, again, generate great data that can give us better insights into understanding this information transfer, right, from RNA to then the processing of the RNA. And so I've spent quite a bit of time in my lab focusing on both computational methods as well as experimental methods that can robustly and scalably identify protein-RNA recognition sites and then which sites are functional, and trying to then derive rules in how RBPs might modulate gene expression. And so, some of the as a grad student, even I had the opportunity to work with Chris, right, to develop a splice site scoring algorithm, MEXANT scan, and to measure the strength of splice sites, right? And that's still being used, I think, by the community and by companies to measure what happens when you mutate a site. Does it actually change the likelihood of it being a splice site anymore? And we've been developing in my own lab at UCSD experimental methods and advancing them, such as enhanced Eclipse, for example, that allows us to probe at very high throughput protein-RNA interactions at near nucleotide level resolution, and all the computational infrastructure underneath it to actually make sense of the data. We've recently developed the STAMP protocol that allows us to fuse different RNA binding proteins to APOBEC or RNA editing enzymes. They can read out the location of these RBPs, even at the level of single cells and even at the level of isoform sensitivity using long read sequencing. And sort of underlying a lot of these methods development, and and these are sort of the parental technologies, but there've been now different versions of these technologies to probe, say, microRNA, biology, or to probe translation, or to look at RNA stability, for example. But underlying all of these are a lot of computational methods to really help us measure if these technologies are actually good and robust, right, and reproducible, and are we actually capturing the information that we want, or are we identifying signals that we didn't anticipate, which is also fine, right? It gives us a new way to study RNA processing, actually. So these are some of the genomic tools that we have developed, and of course we don't do this in a vacuum. We work with many different labs all over the world, right? To sort of enhance the use of our tools, right? To maybe try to disseminate many of these things, right, out there as methods, and derive. I derive great pleasure in hoping, helping the community in this sense. Yeah.
Yeah, 100%. I think you mentioned earlier when you just started, there were probably 13 labs in the whole world studying RBPs, and now your algorithms are being used around the country. And as far as your applications from your lab, it's fantastic to see how that's being integrated. It really building off that with this increased classification and deeper understanding of RBPs, like you mentioned. How are these proteins evolving the paradigm of potential applications in the real world?
Right. So that's a great question. We have seen that some of the tools that we've been developing are accelerating the understanding of how individual RBPs are contributing to disease pathogenesis, right? And by doing so, we are, as a field, much more confident in nominating some of these RNA-binding proteins as therapeutic targets. That's one of the sort of real-world use cases, right? The other perspective is that we're also unearthing new paradigms of how some diseases are actually initiating and amplifying, and perhaps the paradigm of how some of these different diseases are studied are popular, but may not be the most may not be the complete picture, right? And so we're starting to see that there are RNA binding protein and RNA angles at different diseases, thought to be dominated by different mechanisms of action and origin for decades before, but now we're starting to see that there are RNA perspectives in here, and that's, I think, very exciting. We're starting to see that some proteins, for example, thought to be transcription factors are actually RNA binding proteins. Others are identifying that thought to be well-described diseases have an RNA component that is now really an RNA binding protein component, and mapping all these RBP RNA types are also giving us a new resource to then rationally develop ways to interrupt an RBP RNA interaction sites, right? And to develop faster ways to modulate gene expression from both a research perspective, but also a therapeutic perspective.
Fantastic. And really, speaking of applications, I want to pass it off to our second topic here with Chris, really centering around RBPs as regulators, targets, and therapies. Chris?
Oh, thank you, Drew. As mentioned, let's dive a little bit deeper into this RNA regulatory layer and describe the potential of RBPs. So, Gene, as you mentioned, and through the approaches in the conversation thus far, your lab has really revealed that RBP binding sites and their function, as well as their binding preferences, the subcellular localization of RBPs in vitro and vivo, this understanding allows us to consider RBPs as regulators, as targets, and as potential therapies. So let's take a step back and think about this from the very beginning, especially as we think about cellular homeostasis. How do we consider RBPs as regulators in RNA processing?
Great question. This is one of my most interesting, topics I'm most interested in, right? Because in cells, right, you have what I call actually, let me rewind on that, right? So can I just keep? Okay, so you can think of cells as going from one identity to another identity, and for the most part, we at least from the biology field, we think of DNA binding proteins or transcription factors that are involved in pushing cells from say a stem cell to a neuron, right? And there are some RNA binding proteins that also play some of these roles, right? But what I think RBPs are really good at is to maintain cells in that specific state. And so when you have say a neuron and you require the neuron to be healthy and to function properly, to transmit information accurately, RNA binding proteins play roles in localized translation, in moving RNAs from the nucleus to the synapse, right? And to maintain the normal health of the cell, right? The normal function of the cell. And so when you disrupt RBP interactions, right, in healthy, otherwise healthy cells, you may disrupt the balance of RNA localization. You may disrupt the balance of RNA, import, export, and RNA splicing, for example, in the nucleus. And so this leads to cells eventually falling apart, right? And so I think of RBPs as maintaining the healthy state of the cell, crucial for the cell responding to external signals, and sort of integrating that information and enabling the cell to respond to external signals, but also internal networks, for example, that enable the cell to always be in a homeostatic balance, right? And so that's a very important part of what RBPs do.
And as we think about that, you were talking about cellular disruption and things like external environmental stimuli. How do RBPs change in their expression and respond to this disruption? Things like cellular stress, maybe viral infection, to combat that and try to maintain homeostasis.
Right. So one of the most sort of fascinating areas the last 5-6 years now is when many researchers, including our lab, when you look at cells and you start stressing them, right? And you can think of stressors from the most simple, like let's just heat the cell a little bit more, right? Or you add oxidative reagents, or you change add molecules that affect translation. Cells don't like it, right? As they shouldn't, right? And oftentimes they start forming RBP RNA aggregates, right, in the cytoplasm, for example. These are called stress granules. And it's interesting because our RNA binding proteins change their localization, sometimes from the nucleus to the cytoplasm, or they change from a more diffuse state in the cytoplasm into these little condensed foci. And so you can immediately see this response, right, over 30 seconds to minutes, and you can see this cells responding already by changing the protein-RNA RBP interactions and localization. And by doing so, cells are now prepared, right, to survive the stress, right, and respond to the stress in a more focused manner by then allowing the expression of some genes and preventing the expression of other genes, right, and translation of other genes. So it's a very good example of how all cells require RBP interactions to respond to what would be a host of stresses, which would include virus infection and physical damage and so on. So that's a great example. Great question. Thank you.
Absolutely, and thank you for guiding us through it. And I think that also leads well as we take a step forward now, and we talked about a transition from RBPs as regulators to RBPs as drug targets. How do RBPs give rise to disease-relevant phenotypes when they're altered, and how should we really start to think about them in that regard as potential drug targets as well?
Right. So we started some of our projects in the oncology area maybe 6-7 years ago in my group, and it was really driven by studies in the field that have shown that you can modulate RNA splicing to create vulnerabilities, right, in some oncogenic states, with the idea that maybe cells require more processive RNA splicing because there's a lot of RNAs in the cell, and if you can target these splicing factors, maybe the cancer cells would not survive, right? So leveraging sort of building our own research from that perspective, we say, well, actually, this should be true for a host of RNA binding proteins across many life cycle steps, not just RNA splicing, for example, right? And so we started doing CRISPR-based genetic screens to identify, are there other RNA binding proteins and RNA pathways that are safeguard mechanisms for cancer cells to survive, right? Because you need RBPs to provide homeostasis. That's also true in cancer cells. And so it turns out there are many RBPs that would be vulnerabilities in a specific oncogenic context. And there are different RBPs in different oncogenic contexts that are vulnerabilities, right? And so we got really excited because this is like a different way of understanding the oncogenic state, right? And also give us ways to probe and target, candidates to target that might cause therapeutic vulnerability in these different oncogenic states. And so now we are very interested in RNA binding proteins as potential drug targets, right? Again, not a novel concept. Many other groups have looked at proteins, like these specific examples like QR and others, as potential drug targets in a variety of different cancers. But what we're seeing is that there's a host of these. It wasn't just a few that the field had looked at over the past decade, but there are many RBPs that are very good, or important for modulating homeostasis in the oncogenic state. And this, this concept pertains to likely immunology as well as neurodegeneration and many other areas. And so we were actively trying to understand if RBPs play other roles like that, where we want maybe to strengthen their interactions to provide a more robust state for, say, cardiomyopathies, for example. So very excited in the idea that many of the RBPs themselves could be good drug targets. And just to point out, I think the field also have thought that RBPs are maybe hard to drug. I think this notion is rapidly changing over time. I think that like every large class of proteins, there are ones which you cannot drug and ones that you can. And I think the goal is to find which ones are both important in altering the disease state, but also can be drugged, right?
No, agreed. And as actually you mentioned this, you're talking about how different RBPs act as modulators in concert. Do you think it's going to end up being important to target multiple simultaneously to have a disease-modifying effect? Or do you think we'll be able to get away with maybe figuring out which are the really key RBPs and then going from there, manipulating and minimizing the stress granules or the phase condensates, as you talked about earlier?
Yeah. So that's also a very good question. In our lab, we are focusing on identifying the key individual ones that you can manipulate to get a system-wide effect. There might be cases where ultimately you have to modulate a couple at the same time, although that's therapeutically less tractable, clearly, from a development perspective, right? But that may be where data could lead us. So now we don't know that yet. Right now, we've been fortunate that we've been able to identify single targets in specific contexts that are amenable to altering the phenotype of these different cells. Sorry.
Perfect. Okay. Well, thank you for giving us that overview. And now you mentioned this, and I want to focus a little bit more specifically in the CNS. Your lab, in addition to its oncology work, has also proven that defects in RNA regulation in the nervous system can lead to neurodevelopmental dysfunction, things like autism and neurodevelopmental disorders like ALS. And so to elucidate these RBP interactions from the transcriptome and discover potential targets, your lab has spent, at this point, probably almost a decade, pioneering and utilizing single-cell RNA-seq techniques. And you recently developed single-cell approaches to identify these RBP and RNA networks. Can you tell us a little bit more about this approach, share some of the insights, and dive a little bit deeper into the link between RBPs and these neurodevelopmental disorders?
Yeah, a great question. We've been very fascinated about how in different genetic diseases you may have an RNA component, and that component may be different in different developmental stages of an individual, right? And so one of our favorite projects was to look at myotonic dystrophy. And initially we started this in adults, where we were thinking, and we knew from the field, from pioneering work by many labs, Charles Staunton, Maurice Swanson, and others, that RNA repeats in the UTR of genes like the DMPK gene lead to sequestering of RNA binding proteins in muscle cells, and therefore affecting RNA splicing and then leading to muscle dysfunction and weakening. But we started looking at what happens when you have a model of a developing brain, not muscle, in utero, right? Not in the adult. And so we started using iPS-derived cortical organoids, right, as a 3D model to look at neurodevelopment and function from iPS cells derived from myotonic dystrophy patients with exceedingly long repeats that are a hallmark of congenital myotonic dystrophy. And we found that you sequester or affect different proteins, and that leads to, turns out, a loss of function of protein that we is different from the adult muscle case, and then we found that some of that led to a convergence in molecular pathways and phenotypes with a different neurodevelopmental disease in the autism spectrum disorder called Rett syndrome. And so we started trying to converge and make generate new insight into the molecular understanding of these different diseases, right? And as part of the process, we realized that the understanding the cell type specificity of a dysfunction of, say, an RNA disease or RBP, modulation, it's very critical, right? And so, as we were working on this sort of way, this path of inquiry with others in the lab that were developing new technologies to map RBP RNA interactions, and we were very inspired by work from other labs that were able to fuse enzymes to RBPs in the fly genome and fly cells to map RBP interactions. And we said, well, can we fuse different enzymes, one may argue more powerful enzymes, to human RBPs, and we developed the technology that could now, from RNA editing, tell us where RBP is bound. But then we realized that because we can use sequencing as an output rather than a antibody-based immunoprecipitation to identify RNA targets, this platform could be amenable quite easily to any of the available sequencing technologies, including long read technology from PacBio or Nanopore, or the 10x single cell isolation platform, right? And so immediately we could see now, and we had published this, like you can for the first time identify RNA networks, right, at single cell level resolution, and we're so excited to leverage this platform, and hope others do as well, to look at RBP RNA dysfunction at single cell level resolution in complex cell types like organoids, or even in vivo, model organisms, right? And hopefully that tells us that that these interactions that cause dysfunction may be very cell type specific, that may give us better ways to develop safer therapeutics, or gain better understanding of disease progression at stages that we didn't anticipate, right, for these patients.
No, that is a phenomenal overview. Thank you, Professor. And I think as we think about these applications, especially of the information you're able to elucidate, that's a great transition to talk a little bit more about academic entrepreneurship and the Institute for Genomic Medicine. So I'll pass it back to Drew to discuss our final topic.
Fantastic, Chris. Thank you so much. Building on this discussion of novel RNA therapeutics, we would love to learn more about the companies you founded to translate your lab's research in the clinic for patients. Thus far, you've founded 4 biotech companies, including Locana, Eclipse, Bioinnovations, and Zerna and Protanoa. Locana Bio is pioneering a new class of targeted one-time gene therapies that correct dysfunctional RNA and so address the underlying RNA defects in patients suffering from rare neuromuscular and neurodegenerative and retinal diseases. Can you give us an intro to the company and share
Sure.
the launch of the Locana? Any
Yeah,
stories?
yes, of course. So in 2016, my graduate student Dave Nellis and a colleague of mine from the Rady School of Management at UCSD, Richard Steinberg, and I co-founded this company called Locana Bio, really from insights that we had gained in leveraging the CRISPR systems to target RNA, not DNA. And the idea there was that the RNA molecules, right, at that point, the repeat RNA diseases could be amenable to direct interaction to get you allele-specific modulatory effects. And we envisioned a broad platform that we started with CRISPR, but we had quickly evolved into different effector proteins and effector RNAs that could bind RNA specifically. We envisioned packaging them into AAV, and these systems would be able to be a one-time modulation of disease, RNA alleles, right? And so that we built that, incubated that at J Labs in San Diego, and it matured, and it has a large group of people now actively developing research in this area, development and manufacturing of these reagents. And so pretty excited to be part of that journey, and continuing to be part of it as I watch the maturation of the company. Yeah.
Fantastic. And from the other side of the coin here, Eclipse Bio is a different company by all means. It's not a therapeutics one, but genomics kits and services. Can you tell us about Eclipse Bio and give a quick introduction here?
Yeah, of course. So Eclipse Bio was started around the same time as Locana Bio, with the idea that we wanted to raise all boats, right? To raise the tide for all boats in some sense, and we envisioned then, which is even more true today, that RNA therapeutics is going to be an emerging and growing area of interest for biotech and pharma companies. But we also recognize that the entire field of RNA therapeutics required robust technologies and initially genomic technologies, but we also want to move into different areas. And so we envisioned that can we build a leading RNA tools and RNA genomics company that itself wouldn't be a therapeutic company, but would enable all all RNA therapeutic companies, right? And that's also been a very exciting journey to watch, especially with the excitement now in the RNA vaccine space, right? And mRNA as a drug, and also small molecule targeting of RNA. Eclipse has been a very busy company in helping to provide better tools to measure translation, to measure RBP RNA interactions, to measure RNA structure, and to help the community, and both academic and other companies, make better sense of their data, right? And first of all, create better data and then make better sense of that data with novel analytic tools as well. So I've been very excited and proud to see the maturation of Eclipse Bio as well. Yeah.
As we are as well. Taking things a step back, how do you think about crafting an entrepreneurial culture at your own lab at UCSD?
I'm a strong believer of giving intellectual space to my trainees, right? I want them to own their projects, right, intellectually, right? And they, so we try to provide an environment where they really dive in and build a deep understanding of a specific area, and where they can recognize what are the key big problems, and they try to develop ways to solve some of these big problems. But at the same time, we balance this out with a, I think, pragmatic and sober view of like how some of these research fundamental findings can be helpful to patients or to the community, right, to enable others. Like we have a very community-driven sort of spirit in my group, and so I just sort of creating this sort of ecosystem of if you can probe and do great science, we can help envision how this great science can be translated. And we have many entrepreneurs that have come from my lab, but also want to work with my group, right? And from different aspects, and we are very fortunate to have this environment here in San Diego that has allowed us to do that. And a lot of my students and trainees are very entrepreneurial, and they come with this mindset more and more frequently, right? Where they want to do something like bleeding edge, make fundamental discoveries, but hope that they can apply these discoveries to a wider community. I just give them the space to do that. I try to give them resources to do that, and the mentorship and the guidance to help them make the right decisions. And sometimes they listen to me, sometimes they don't, but everyone has the ability to make their own mistakes, right? And so we give them a safety net to try to catch them also when mistakes are made, are made individually or as a group. So
no, 100%. That's, we always love to see how entrepreneurial cultures are formed, and always try to get the tidbits and from folks who have done it on their own and maybe have a few battle scars on their own side. As we speak about other labs and cultures, really, do you have any advice to young PIs seeking to translate work of their own from academia to industry?
I'm not sure I have the most sage advice here. I think a lot of it is driven by trying to be very honest about where the research is, at what stage, and at the same time you have to balance that with aspirational optimism of where things could be. And there's a hard balance. I think sometimes I admit we sometimes start things too early, sometimes we wait too long and start things too late, and so it's a balancing act, right? And I think my advice to young PIs that are entrepreneurial and want to be entrepreneurial is to really build a very strong team around you, not only of talented technical individuals, but really well-aligned individuals in terms of culture and a balance of pragmatism and idealism, right? And then try to, I always realize we can't do everything on our own, so we try to recruit and bring in friends and colleagues that are like-minded. And so I think build a village around you, and I think it's more fun that way. And I think people are more excited to work with each other to pursue a common goal. I think the idea that academia has to be a lonely path and you are each doing your individual project, I think that's not. I don't see it that way. I sort of encourage my team to move together as a group. Maybe my training in the Navy has helped that, but on the ship you can't. No one can operate a ship on their own, right? You need a whole host of different skill sets and perspectives.
I love that focus on team, and I think that's a great way to maybe focus on bring the episode to a close here, Gene. So before we do come to a close, just one rapid fire question. Any closing thoughts or shameless plugs? How can our audience learn more about your work?
Yeah, so you can go to our lab website. I think it's yourlab.com, but you can also just email me, and we're always looking for, again, like-minded individuals that are interested in like RNA biology or science in general, and entrepreneurs that want to work with us. That's always been fun for me to look at how to build amazing teams to try to pursue interesting goals, right?
All right. Thank you, Gene, for an absolutely fantastic episode. I know I have a lot more questions.
Thank you, Chris.
So we'll
Thank you, Drew.
Thank you again.
Yeah, you too. Take care. Bye-bye.